Hello, my name is Garik Mukherjee, and for my biology project, I'm gonna extract the DNA from the strawberries. So for that, I need the strawberries, of course, 1/3 cup of water, salt, the dishing washing solution, uh, chill freeze isopropyl alcohol, and some extras such as the cups that will act as my test tubes, and also the cheesecloths that will gonna help us for our separations from different reactants. So first we gonna make our extraction solution. So we need half a tablespoon of salt, half a tea spoon of salt, and of course 1 tablespoon of the dishwashing liquid, and mix it until it's homogeneous. So what this is good for, the dishwashing soap has a surfactant molecule which helps to break down the cells, the cell membrane, and break into until the nucleus isolate the enzymes or the proteins, so we can extract the DNA. It also eliminates other proteins or enzymes that are responsible for dissecting the DNA, while the salt is creating the ionic solutions, so it helps that to neutralize our DNA and easier all the DNA molecules to clumps together so to be visible in our eyes. So this is our extraction solution. So we'll take our cheesecloth and cut it at a certain get the scissors. So for our cheesecloths, we're gonna just rub it, which acts as a membrane to allow certain solutions, certain molecules, parts of our solution to pass through, and we wrap around our funnel. I think this kind of makes horrible. I apologize for that. Okay, so for right now, let's take let's say 3 strawberries. Let's remove this green part, which is not needed. That's 1. That's 2. And that's 3. And we will place it into our plastic bag. Okay. And let's remove all the air, like maybe around like 2 minutes, about 2 minutes. Squeeze it until it depends how strong you are, of course. Just squeeze enough until you have very liquidy solution all the big parts are removed. Because the more the surface area, the smaller the size is, the higher surface area, the easier that for us to able all these molecules to react to each other, able to dissolve it, which makes us to dissolve, to extract our DNA at the more efficient. Well, we expect it just like this till we get. I think this is no. I don't know if you can. Let's make it over. Okay. Okay, I think this is great enough. Okay, now we're gonna need the 3 tablespoons of our extract solution. So to add it, so we're gonna place it here, and that's 1, that's 2, and that's 3. Again, remove our air and mix them. So now that our all those reagents, whether for dissecting the cellular membrane, isolating the DNA, makes the DNA clumsy, and etc., are all happening right now. Okay, about like mix it. Okay, I think we can see the difference. Now let's put our tablecloths, funnel and the tablecloths, and pour everything here. So I'll put a separate cup which acts as my too. And now let's pour everything here. Okay, let's deal with this one. Okay, now let's add the rest of it. Okay, I think that's all of it, and we're just squeezing. This is kind of messy, but I think this works. Okay, so this is that. Okay, let's use a tissue to clean ourselves. Okay, so we have here, so there are too much foam, like foam, so let's separate the foam. So let's separate the phone. Let's remove all of these. So the ones that we don't need. Okay, there's some phones that are happening here. So so this is what we have so far. Now we're gonna use the rubbing alcohol, which actually separates the DNA, because this is consists of many like organic solvents and a very tiny DNA. So we're gonna use the aqueous layer, which is our alcohol. So our DNA will separate it and be more in aqueous layer due to its structure. So in that case, let's transfer our in a small cup. Okay, so this is all what we have. Now about same height, let's add our alcohol. Okay, very slowly. Okay. Now slowly we can see, I don't know, this is visible, but we can see the DNA actually getting extracted. So let it sit by a few minutes until it extracted. So here, as soon as you see that they are slowly extracting, I will use a toothpicks and try to remove, but there are some white drops that you might be able to see, which shows that it's our DNA clumps together. Let me see if I can use the toothpicks and Opa, see, that's our DNA right there. So we got able, there are still more that I can try and extract it out. Okay, let's see, there's Opa, and that's our DNA for strawberry.